Hello everyone, welcome to Life Science Insider. In this video, we will be telling about how to reculture or passage the cancer cells. Okay, so here we have taken out the T flask from the incubator, CO2 incubator, and we are checking in the inverted microscope. So here the confluency is great, and now it's time to reculture it or split it into the one flask will be split into a two flask depending on how you want to split it. So here a lot of dense cells you can see. So this is how you have to manage this, and if there is more than, like, it is approximately 80 to 90% confluency, then it is considered to be recultured. So this biosafety cabinet has been run for 15 minutes of UV sterilization. After that we are only using it. So we are taking out all our reagents, and we have already wiped the floor of the biosafety cabinet, and this is the CO2 incubator, and we are taking out our cell to for the reculture. So it has approximately 90% confluency, and if we waited more, then it will start clumping, and it will grow over each other because they don't have the contact inhibition. So first of all, we have taken out from the incubator, and we will discard the previous media. Okay, now it is empty and the cells are adhered into the surface of the T flask. Make sure you should not touch the any of the things are there from the edge of the bottle. So the thing is, okay, use the pipette, that will be the 1 ml, and what you have to do, you have thrown out the discarded the previous media. Now wash it with the PBS, that is a phosphate buffer saline. It must have the pH of 7.4. So we are doing this because we want to wash most of the debris. They, like, some of the cells may be died and their dead bodies will be there, and we have to discard it. So we will wash this media and the debris with the 2 ml of the FBS, and we are using this T flask, that is the 25 centimeter square. We also call this T25. Okay, so we will put the 2 ml of the PBS here. Okay, not FBS, PBS, that is phosphate buffer saline, and we will gently move it so it will spread all over the surface of the cells. So whatever the debris or dead cells are there, it will come into the FBS, and the adhere cell will be adhered there. Okay, and again we have to discard it. So we have discarded it, and now what we have to do, we will trypsinize it. Trypsin is a serine protease. What is do, it it degrades the cell addition molecules of the cells from the matrix. So it takes out the cell out of their attachments. Okay, so we will take out the, we are using 1 ml of the trypsin. Okay, it is a Trips Express, it's a recombinant enzyme, it's milder than the trypsin. So we will add the 1 ml of trypsin here, and we will gently tap it. So it will the trypsin will start working, and we will gently tap it so it will lose its adherence. Okay, so this is how we have to do it. Now like, we tap for the 10 to 15 seconds, because this trypsin will lose the adherence of the cells, so these cells will come in the solution. Okay, we will put in the incubator for 1 minute at the 37 degrees Celsius. This that is called as the warm trypsinization. So it will increase the rate of trypsin reaction, so cell will disadhere or dissociate from the surface. And then what we have done, we have taken out after the 1 minute, and again we tapped it. So now what we are going to do, we will add the 4 ml of working media that contains the fetal bovine serum. This fetal bovine serum will neutralize the trypsin. Make sure whenever if something falls from your hand and in the floor, don't use it, just throw it. And make sure your the edge of the tip should not touch anything. Okay, so you have added the 4 ml of working media that contains the ABS that neutralizes the trypsin. Now again we will tap it, so most of the remaining cells will get out of its attachments. Now we will take the 15 ml falcon tube. We will take out all the media that contains the disadherent cells. Now we will put it into the 15 ml of falcon tube. So it will be almost 4 ml because we have added the 4 ml media, and this media is containing, that is in the flask containing what, this cells. Okay, so you can see that it has milky appearance. That means the cell has been decided and mixed with the media. So now you will take out all the media that contains the cell and you will add into the 15 ml of falcon tube. Make sure your thoroughly mix at the last because it may contain a lot of number of cells. Now put that falcon tube into the centrifuge machine, balancing the counter, it having the same weight, and close it properly because it should be opposite of that. Close it properly. Close the lid and it will close by itself, and you have to start. And what will be the settings for this? It will be 5 minutes at 1200 rpm at 25 degrees Celsius. Now it is been completed after 5 minutes. Take out properly. Take out your falcon tube out of the centrifuge machine. See it if it have the palette, and it should be visible. Now you can see the palette will be in the one side. Here you can see the palette is in this side. Now discard the media from the opposite side of the palette. So this supernatant will be discarded and the cells will be attached in the bottom of the falcon tube. Now what we have to do, we will add 2 ml of the phosphate buffer saline to wash any kind of debris that is present with the cells. Now you are taking out the 2 ml of PBS and putting into your falcon tube which having the cells. Now you will close it, you will mix it thoroughly by tapping also. Now again you have to do the centrifuge with the same settings, that was 5 minutes, 1200 rpm, 25 degrees Celsius. Meanwhile, you can take out the new tea flask where you will be splitting one flask into two. So you have to add the cell line name and the date when you have passaged and the numbering of passage. If you are splitting for the fourth time, you will write 4 passage and the flask number. Do the same thing for the second flask. So now we are done with the centrifuge with the cell containing PBS of 2 ml and we are taking it out. Okay, we have to check the palette if it is there. Now from the opposite side you have to discard your PBS and the cells will be adhered into the bottom. Open all your new tea flasks where you are going to split the cells. Add 4 ml new working media into the falcon tube. Now mix it well with the help of the pipette by aspiring and dispersing it, and it should be mixed with the current media also, and it should appear milky. The pellet should not be visible now, it should be mixed with the media. Now add 2 ml of the media containing cells from the falcon tube into the tea flasks. 2 ml in one, 2 ml in another one. So that means you have spent all the 4 ml that you have mixed into the falcon tube. So now you have added the 2 ml in the one tea flask and 2 ml in another flask. Now you can add 3 ml of media from the fresh media into each new flask, and make sure after every and each process you have to change the tip, because you should not take any risk of contamination. Now you have to firmly twist and turn the flask. Make sure it should be in the horizontal frame, but it should cover all the surface of the tea flask. You can also do both of them at the same time. Now these navally cultured flasks will be stored in the CO2 incubator, and it is mandatory to check the confluency every day, and if the confluency is reached is more than 90%, it's more than 18%, you should reculture it, because you should not take the risk, and next day you will check. Thank you so much guys for watching this video, and please subscribe this channel so you can learn new techniques related to the biology, including the cancer cell research. Thank you.